presentation of the titled Circulating Tumor Cells-driven Choice of First-Line Therapy for ER-positive, HER2-negative metastatic breast cancer, overall survival analysis of the randomized SITC Circulating Tumor Cell Trial, presented by Dr. François Clement Bidart.
So thank you very much. Good morning to everyone. So this is the long-term analysis of a trial that has been previously reported, like at this Congress a few years ago, and now it's about overall survival result of this strategy trial. So it was an academic trial, and so these clashes. So the idea, all of the trial is about the choice between chemotherapy versus endocrine therapy in patients with ER plus HER2 negative metastatic breast cancer, because we have guidelines that are telling us that endocrine therapy is the preferred therapy and must be considered, must be prioritized. And this is mostly because there is few side effects. But we also have in guidelines the option of prescribing chemotherapy for patients with aggressive disease and unfavorable prognosis. So there is no validated decision-making algorithm, and this is leading to highly heterogeneous treatment decisions between countries, but also between centers and also between physicians in the same center. So what we are using now, what we have used in the STIC, is a biomarker, which is the count of circulating tumor cell by the cell search technique. And so we know it is standardized, it is FDA cleared, it could be run in CLIA labs, so that is very strong. And we and others also reported that a high CTC count is a super strong adverse prognostic factor. So it really tells that a patient with metastatic breast cancer will have a poor prognosis, both in terms of PFS, but mostly in terms of overall survival. So we know it has already been shown that it can complement and not duplicates the other clinical pathological factors. So the concept of the STIC CTC, so again an academic conceptual trial, is to use the CTC count as an aid to drive the treatment decision between endocrine therapy for, let's say, slowly evolving disease versus chemotherapy for more aggressive disease. So something that would be an objective assessment of whether the breast cancer, metastatic breast cancer is aggressive or not, and then to optimize the treatment choice. So this is the design. In the standard arm, a patient received either endocrine therapy or chemotherapy according to the investigator choice. The CTC count was collected, but remained blinded to investigators. So in standard arm, we are following the treatment decision by investigators between either CTC or chemotherapy, either endocrine therapy or chemotherapy. CTC arm, we dismissed the clinical choice, and patients were treated according to the CTC count. Low CTC count means patient had to be treated with endocrine therapy. High CTC count patient had to be treated with chemotherapy. Okay, so we have 4 subgroups, and clinical low subgroup are the patients that would have been treated with endocrine therapy by the physician, and so clinical high treated with chemotherapy according to the physician. And so you see we have 4 subgroups in our trial population. So clinical low, CTC low, this patient receives the same treatment because in this patient population, the CTC count confirmed that endocrine therapy should be given as a first-line treatment. On the bottom part of the slide, we have the CTC high, clinical high patient. So this patient received chemotherapy in both arms. What is mostly interesting is the 2 groups with discordant estimate. So clinical low CTC high patients, this patient received endocrine therapy according to the investigator who were actually following guidelines. And because of this high CTC count, patient received chemotherapy if allocated in the CTC arm, and vice versa for the clinical high CTC low subgroup. So more than 750 patients enrolled at 17 sites in France. The primary objective, the short-term objective, was already reported and was achieved with flying colors and published in JAMA Oncology in 2021. And here in this final analysis, it is about the overall survival, which is the long-term and the most important readout for metastatic breast cancer. So I'm just here, brief slide, just to say that when patients were allocated to endocrine therapy, they mostly in our trial received single-agent aromatase inhibitor or fulvestrant. And CDK4/6 were not approved at time of accrual, so which means it's not a first-line trial in the CDK4/6 era, but still we were able to find that 42% of our patients received CDK4/6 inhibitors as a later line of treatment. Patients treated with chemotherapy received mostly paclitaxel and capecitabine. So if you look at overall the distribution of the patient between the clinical choice and the CTC count, you can see that we have 60% of patients with concordant estimate. Clinical low, CTC low, clinical high, CTC high. So in this 60% of patients, CTC count confirmed the clinical choice, which is good. But we have this 40% of patients with discordant estimate and with different treatments. So this is the most important result we are reporting today. So it's a group with discordant estimates, so clinical low, CTC high patients. So 1 patient out of 4, so 25% of patients. So were treated according to guidelines by endocrine therapy by the investigators, and because of a high CTC count, they receive chemotherapy if allocated in the CTC arm. You can see that we have not only a statistically and clinically significant survival benefit in terms of OS, but also in terms of PFS, but also in terms of OS. So you see the delta in median overall survival in favor of chemotherapy in this patient group is 16 months, which is quite relevant for the patient, I think. So what about the other discordant subgroup? In this subgroup, clinicians were proposing chemotherapy to patients, but these patients may have received endocrine therapy because of a low CTC count if they were allocated to the CTC arm. And here, so we are showing no survival benefit of chemotherapy over endocrine therapy. So which means in these patients, the standard of care was, according to guidelines, was probably endocrine therapy and must remain endocrine therapy because we have no superiority of chemotherapy in these patients. So patient with concordant estimates, so we have, of course, similar outcomes, and the survival by arm in the overall population is very similar between the two arms because, again, of these 60% of patients in whom CTC count confirms a clinician choice. As to conclude, so that STIC CTC is the first trial to demonstrate the clinical utility of circulating tumor cell count in metastatic breast cancer with a high level of evidence. CTC count confirms the allocated therapy, so confirms the clinician choice in about 60% of patients, but there was also 40% of patients in whom patient was changed. We have a major benefit, so which means CTC count is able to pick patients that deserve to be treated with chemotherapy rather than following the guidelines and giving endocrine therapy to all patients. Main limitation, the trial was not run with CDK4/6 inhibitor in first line. And here I have to be really strict in my advice when you will report this study. It is not about selecting for endocrine therapy plus CDK4/6 versus chemo. That is not the case, so our trial do not apply to that situation. So the standard of care for CDK4/6 inhibitor naive patient must remain endocrine therapy plus CDK4/6. However, having said that, we still have a huge dilemma in the clinics to choose between endocrine therapy and chemotherapy after a patient has progressed on CDK4/6 inhibitors. And we also know that these drugs are moving to the adjuvant setting now, so which means we believe that CTC count is opening new opportunities in terms of to help the treatment decision in patients who received previously CDK4/6 inhibitors and for whom we have short PFS with endocrine therapy and vectorized chemotherapy, so ADC, is very likely to become a very attractive option in the future. So which means current standard of care, which is to keep on with endocrine therapy for many lines or as much as possible, will be heavily challenged with these ADC coming in first line. And this might be a part of the many biomarkers we could use to optimize the treatment decision. Thank you.
Thank you. Questions for Dr. Bedard?
Carolyn Helwig, ASCO Post. So we've been hearing about CTC counts for like a decade or more. And I just wonder, how are doctors, have they adopted this in the community? And maybe, I know you're French, but maybe Dr. Arteaga. It seems like it would be useful, but I don't know how much it's being used.
patients are asking for it, and some physicians are using
Patients
this
are asking
information.
for it.
Yeah, patients ask for it. Not all of them, but some do.
So
But
if-
without asking for it, are doctors ordering
No,
these
these
tests?
things are discussed also, but so maybe you can comment more on that.
Yeah, if I may.
Sure.
So primary development of CTC was to assess early the efficacy of a drug, the changes during therapy. So you had to repeat and to do it twice.
Right, okay.
And then if the treatment was not working, you would have to switch to another treatment. And that strategy so far has failed. It's failed for CTC, and for ctDNA, we don't know. So we don't really know, there is utility there. Here it's something very different as an approach. It's a standalone. You do it once to choose before treatment between different treatments. So let's say it's a bit unique trial in this setting. And when we reported PFS, everyone was saying, PFS is good, but we need to wait for OS. And of course, OS is long read out. So that trial was initiated a long time ago. But it takes time to mature and to get a good level of evidence. So.
Okay.
So, Dr. Partridge, do you use
Yeah,
a
what's
lot
your
of
thoughts?
CTCs?
Sure. So I think that it's not been widely adopted in part because these are new data, A, and B, there are other tests that people can also consider.
the mic is
I don't think your mic's on.
Oh, sorry. It's not so clear that it's any better at this point, or has been any better up until now, than some of the other tests that you can do, including intrinsic subtyping on the metastatic biopsy or other parameters.
Okay, thank you.
Liam Davenport from Medscape. So obviously, in a way, I guess ideally you would like to do this again, but in the context of CD46.
No, I would not, because let's say if we were to run after all changes in treatment, we would do that with AI and CDK, but I'm not sure we can challenge. Clearly, this test is interesting when you really don't know. We know that chemotherapy yields to a limited, toxicity. We have toxicity, it's costly, and the PFS with chemotherapy is no more than probably 1 year. And we know the PFS with CDK4/6 inhibitors is super long. So again, I'm not sure that it has a role to play, like in a choice between chemotherapy and CDK4/6 inhibitor plus endocrine therapy. And also, if we were to run that trial, when the trial will read out with PFS, it would say we have to wait for OS. And when the trial will read out for OS, then it will be no longer about AI plus CDK. It might be new cells, and people will say, obsolete, and so on. So it's more about, I think it's, again, more in endocrine-resistant patients. The concept should stay, and to find out aggressive disease and with short PFS. And these patients should be treated probably with chemotherapy. And that is a subject that will become a very hot subject in the coming years because of, namely, NR2 and other ADCs that are coming that will push a lot and with good data to do chemotherapy quite quickly in the sequence. And we'll have to triage. So.
And just one other thing I wanted to ask about is there's quite big discordance, 40% discordance. I wondered if you could just speak to that about the difference between the guideline recommendations and what your CTC count showed.
Yes, so let's say we have guidelines, but we know there are, let's say, the application of these guidelines is somehow heterogeneous. So that's not surprising that we have quite a high rate of chemotherapy proposed by investigators. Guidelines in our countries are telling us to do endocrine therapy. I think now it's very commonly accepted in first line with CDK4/6 again. But there's still this idea of, maybe endocrine therapy is not beneficial to all. And I think here, it's the first trial that is able to pick a population in which chemotherapy is really beneficial. So that's a quite a population. So it may worth, pay for the test just one time. And if you can find one patient out of 4, which could derive the major benefit.
Yeah. Any questions in the webcast?
Yes, I have 2 from Charles Bankhead with MedPage Today. Do you have any data or evidence to show how use of CTCs to guide treatment decisions compares with genomic assays that have been used to guide treatment decisions? Is the first question.
So I think that the level of evidence supporting the clinical utility of RNA-based assay in the metastatic setting is very low. So we have no trials that demonstrated. We have data showing that these mRNA-based signatures have prognostic implications. So that is called what we call clinical validity. We can also question the level of evidence supporting this clinical validity, but there is absolutely no data in favor of clinical utility. This is a clinical utility trial, so it means like it's completely the next step. Clinical validity is really easy to report. Clinical utility, you need a prospective trial in which you compare the different strategy in a prospective manner, in a randomized manner, so that's not the same level of evidence for sure.
Next question.
He says there's no need. It's been addressed.
Very good. Thank you very much.
So before you go, so in this page, you have not, in this study, did you also collect plasma to look whether the genomic information in that plasma adds to it or even outperforms CTC counts?
Yeah, sure. So that is the main question. Clearly, everyone is about ctDNA, including myself. In this trial, no, we have not.
That's very nice.
That's it.
Okay. Thank you very